Okay, so this session is about mitosis, so specifically cell division and mitosis, and this is from topic 2A on AQA A-level biology. So, yeah, mitosis is a type of cell division, and that's what we're going to look at now, how it works and the processes of it. And basically, the idea of mitosis is that it is a cell division that produces genetically identical cells. So mitosis, there are two types of cell division in eukaryotic cells. You have mitosis and meiosis, and we've looked at meiosis before. If you're not quite sure, go back and have another look at another video that had covered meiosis, sorry. But mitosis is that production of genetically identical cells, which meiosis doesn't do. So in mitosis, a parent cell divides and produces two genetically identical daughter cells. So we call them daughter cells. We refer to them as parent cells and daughter cells. So one parent cell, the original cell splits, it divides, goes through some other stages for it to be able to do that, and produces two genetically identical cells, and we call these cells daughter cells. And they contain an exact copy of the DNA of the parent cell. So they are both identical to that original cell. And mitosis is needed for the growth of multicellular organisms and for repairing damaged tissues. So that is the point of mitosis. We need new cells all the time. Parts of our body needs new cells for us to grow and to repair damaged tissues. In multicellular organisms, not all cells keep their ability to divide. So some cells don't go through replication later on in life, and some of them just stop. Others need the ability to continue to replicate. So it just depends on the type of cell. But early, when multicellular organisms are growing, mitosis will be occurring. And then once you have a fully grown adult organism, and some of those cells stop dividing. However, some that do continue to divide, they follow a cell cycle, which you can see here on the screen. And mitosis just forms part of this cell cycle. And the cell cycle consists of a period of cell growth, and we call this section G1 or gap phase 1. So sometimes it's gap phase or just G1. So this is G1 here. So this is G1 here, and this is the growth stage. So in this G1 stage, this gap phase 1, cells are going to grow and new organelles and proteins are made. So this is where DNA replication is going to occur. And this process, this period actually of G1, S and G2 is all called interphase. And this is what happens before mitosis. So this is that cell growth, that DNA replication, and then mitosis happens after this. It happens after interphase and is subdivided then into 3 separate growth stages. So these separate growth stages, we've got gap phase 1, and in G1, our cell is going to grow and new organelles and proteins are made. So that's the first part. So our cell's going to grow bigger, new organelles are being made, and new proteins are being made. Then we go through S phase, which is synthesis. And in this part, the cell replicates its DNA ready to divide by mitosis. So that's DNA replication occurring in S. Then we've got G2, which is gap phase 2. And this is growth and preparation for mitosis. So the cells are going to keep growing at this point, and proteins that are needed for cell division are made here. And then we go on to mitosis, and the cell cycle starts and ends in mitosis. So mitosis has 4 division stages. So mitosis is really just one large continuous process, but we describe it as a series of division stages. The actual mitosis stages are prophase, metaphase, anaphase, and telophase. But you'll hear of interphase spoken of as well, and that is the stage that comes before mitosis in the cell cycle. You'll hear IPMAT referred to a lot. It helps you remember the names of the different stages in the correct order. So IPMAT, I for interphase, P for prophase, M for metaphase, A for anaphase, and T for telophase, IPMAT. First, we're going to look at interphase. So though it's not technically a stage of mitosis, it comes before mitosis in cell cycle, and it's important that we understand this part of the cell cycle. So here, in interphase, normal cell functions are being carried out. So during interphase, the cell is doing its normal job, anything else it needs to be doing. But also, alongside it carrying out its normal cell functions, the cell is also preparing to divide. Part of this preparation for division is DNA replication. So the cell's DNA is going to unravel and it's going to replicate to double its genetic content. So this is what's happening during interphase. This is our cells, and then we've got this unraveled DNA, and we've got containing 2 copies of each chromosome. All the organelles inside the cell are going to replicate, so it has spare ones, which will be useful when we're then dividing the cell into making 2 new identical daughter cells. And its ATP content is going to increase. And that's really, really important because ATP provides the energy that is needed for cell division. So first stage of mitosis is prophase. In prophase, the chromosomes condense, and they condense by getting shorter and fatter. So they condense, they get shorter, they get fatter, and we get something called, we get tiny bundles of protein forming. These tiny bundles of protein are called centrioles. And what these centrioles do is they start to move to opposite ends of the cell. And they form, as they're moving to opposite ends of the cell, a network of protein fibers across the cell, and it's called the spindle. The nuclear envelope, which is the membrane around the nucleus, breaks down, and now chromosomes lie free in the cytoplasm. So as prophase, our chromosomes condense, our centrioles move to opposite ends of the cell, and the spindle fibers start to form. Nuclear envelope starts to break down. Now I'm just going to focus in on the words that we're going to use now for chromosomes and during mitosis, because the names change in places. So when mitosis begins, your chromosomes are made up of 2 strands, and these 2 strands are joined in the middle by something called a centromere. So you can see that on both images here. We've got a centromere here in the center of the cell, center of the chromosome, sorry, and we've got the centromere here. Now, the separate strands are called chromatids. So a chromosome are the 2 strands that are joined in the middle. If we separate the 2 strands, they then become a chromatid. And there are 2 strands in a chromosome because each chromosome has already made an identical copy of itself during interphase. So again, I'm going to repeat that just so that it's clear, because this often causes quite a bit of confusion. During interphase, we've already gone through that DNA replication. So we have 2 strands. And chromosomes are made up of 2 strands at this point because each chromosome has already made an identical copy of itself during interphase, and the separate strands, remember, are called chromatids. When mitosis is over, the chromatids end up as 1-strand chromosomes in the daughter cell. So that's just really important to remember. So that's prophase, chromosomes condense, centrioles move to opposite ends of the cell, and our nuclear envelope breaks down. Then we go on to metaphase. In metaphase, the centromeres divide. So those centromeres that are in the middle of those chromosomes, they divide. Skipped ahead, sorry. So we're on metaphase. I've just jumped ahead, and I'm telling you about anaphase. So metaphase, sorry. Your chromosomes, each made up of 2 chromatids, they're going to line up along the middle of the cell, along the cell equator. So they line up with the centromeres there, and they become attached to that spindle that's coming from those centrioles. So centrioles that we had up here, which are those proteins bundles. They have those spindle fibers coming out from them, and the chromosomes become attached to those spindle fibers by their centromere in the middle. So centromeres on spindle equator, and they join up there. So that's metaphase. Chromosomes line up along the middle of the cell, and they attach to the spindle by their centromere. And in anaphase, the centromeres are going to divide. And when the centromeres divide, they separate each pair of sister chromatids. So remember the chromatids when we split, because we've got 2 copies. The spindles contract and they pull the chromatids to opposite poles of the spindle. And they go centromere first. And because they go centromere first, this makes the chromatids appear V-shaped. So if you ever looked at this with a microscope, you'd be able to tell it's going through anaphase because we would have this V shape here. It's really important that you know the appearance of the cells at each stage of mitosis for the exam, because you could get given an image and asked to explain what stage of mitosis it's at, or you could get told you're looking at a cell and it's at the stage of anaphase, explain what anaphase would look like. So that V shape is really important that you mention that. So the centromeres divide, separate each pair of sister chromatids, the spindles contract. And when the spindles contract, that pulls the chromatids to the opposite poles of the spindle, centromere first. And because of that, it causes the chromatids to appear V-shaped. And then we go on to the final phase of mitosis, which is telophase. And in telophase, the chromatids reach the opposite poles on the spindle. Once they are at the opposite poles of the spindle, they begin to uncoil and they become long and thin again. Now that they've become long and thin again and they've uncoiled, they are now called chromosomes again. So we now refer to them as chromosomes again. A nuclear envelope then forms around each group of chromosomes. So we now have 2 nuclei. The cytoplasm divides, and that's called cytokinesis. And there are now 2 daughter cells that are genetically identical to the original cell and to each other. Mitosis is finished, and each daughter cell starts their interphase part of the cell cycle to get ready for the next round of mitosis. So telophase, chromatids get to the opposite poles and uncoil. Nuclear envelope forms around each set of chromosomes. We then get 2 nuclei, our cytoplasm divides, and we get 2 identical daughter cells. So again, IPMAT, interphase, prophase, metaphase, anaphase, telophase. Interphase, normal cell function going on, cell's going to prepare itself for division. To do that, it goes through DNA replication, organelles are replicated, and we have an increase in ATP content. Prophase, our chromosomes condense, they get shorter and they get fatter. Centrioles, which are bundles of protein, form and move to opposite ends of the cell. And when they do this, they form protein fibers across the cell, which we call the spindle. And the nuclear envelope starts to break down. Metaphase, those chromosomes are now going to line up along the middle of the cell, and they're going to attach to the spindle by their centromere. And the spindle fibers, remember, come from those centrioles at either pole of the cell. Anaphase, our centromeres are going to divide, and this separates each pair of sister chromatids. The spindles are going to contract, which pulls the chromatids to opposite poles of the spindle, and they go centromere first, which causes the chromatids to appear V-shaped. And finally, telophase, our chromatids reach opposite ends of the poles and they uncoil and become long and thin again. We now refer to them as chromosomes again. Nuclear envelope is now going to form around each set of chromosomes, and so then we have 2 nuclei. Once this has happened, cytoplasm is going to start to divide, which is called cytokinesis, which then causes us to have now 2 identical daughter cells that are genetically identical to each other and to the parent cell. Now, each stage of mitosis can take varying different amounts of time. And we can calculate this. So I've just put this example on to talk it through with you because you could get asked this in the exam, and we're just going to look at this now. So it varies. Each stage of the cycle varies. So a scientist observes a section of growing tissue under the microscope. He counts 100 cells undergoing mitosis. Of those, 10 cells are in metaphase. One completed cell cycle of the tissue lasts 15 hours. How long do the cells spend in metaphase? And it asks you to give your answer in minutes. So let's look at the information that we've got. So the scientist has observed that 10 out of 100 cells are in metaphase. So this would suggest that the proportion of time the cells spend in metaphase must be 10 hundredths of the cell cycle. You know that the cell cycle in these cells lasts 15 hours. So 15 times 60 would give us 900 minutes because we need to give our answer in minutes. So we need to find 10 hundredths over 900 minutes, which would be 90 minutes. So we would then say that the cells spend 90 minutes in metaphase. Cancer can be the result of uncontrolled cell division. So mitosis and the cell cycle are controlled by genes. And they're not just random processes that just happen, they are controlled by genes. And normally, when cells have divided enough times to make enough new cells, they stop dividing. So they have a process in them that stops them from dividing once they've made enough new cells. We don't just have this replication going on and on and on, and this uncontrolled cell division. Once we've got enough new cells, so once maybe we've healed a wound, or once some old cells or damaged cells have been repaired or we've grown enough, they stop dividing. But if there is a mutation in a gene that controls cell division, the cells can completely grow out of control. The cells will then keep on dividing and they will make more and more cells, which will form a tumour. And as you know, cancer is a tumour that invades surrounding tissue. So uncontrolled cell division so cancer is the result of this uncontrolled cell division. Now, some cancer treatments actually target the cell cycle. So some treatments for cancer are actually designed to control the rate of cell division in tumours, in tumour cells by disrupting that cell cycle. So that cell cycle where we've got interphase and mitosis, they actually go in and they disrupt it. So this kills the tumour cells. The issue with these treatments is they don't distinguish tumour cells from normal cells. So they also kill normal body cells that are dividing. However, they only kill normal body cells that are dividing, not just any body cell. So they're looking for cells that are dividing. And because tumour cells are going to be dividing much more frequently than normal cells, the treatments are more likely to kill tumour cells than normal cells. So some cell cycle targets of cancer treatment include these ones. So we can target G1, that gap phase 1 section. And that's where cell growth and proteins are going to be made. So during this G1, where the cell's growing and proteins are being made, some chemicals such as chemotherapy, some chemical drugs, they can actually prevent the synthesis of enzymes that are needed for DNA replication. So in this process, in G1, normally we've been making those enzymes that we'll need for DNA replication. Chemotherapy can actually stop this synthesis of enzymes that we need for DNA replication. If these enzymes aren't produced, the cell is then unable to enter the synthesis phase, the S phase, that DNA replication phase. That disrupts the cell cycle and then will force the cell to kill itself. Another way we can treat cancers by disrupting the cell cycle is focusing on the S phase, and that's where that DNA replication is occurring. So radiation and some drugs damage DNA. At several points in the cell cycle, and that includes just before and during the synthesis phase, the DNA in the cell is checked for damage. So your body's really, really clever, your cells are clever, they can check for DNA for damage continually. And if there's damage to it, if there's mutations to it, they stop it from growing. They basically will kill the cell, it'll go through apoptosis and the cell will be killed. So what they try and do here is they go in and they purposefully damage the DNA in those cells. And the idea is some mutations can get passed through and some can sneak through. But if we go in and damage it really, really badly, when this cell's DNA will be checked, we've caused severe damage and it's detected, and then the cell will kill itself, and that prevents any further tumour growth. So that's the process and ideas behind that. So a quick look at an exam question here. And like I was saying, you can regularly get asked to identify the stages of mitosis from images, or describe what's happening in stages of mitosis from images, or describe what a cell might look like at different stages of mitosis. So in this one, the diagram shows cells at different stages of mitosis, and you're asked for each stage of, you're asked for each stage of the cells A, B, and C, you're asked to name the stages of mitosis. So looking at A, we've got that lining up of the chromosomes along the middle of the cell. We're going along the middle, which means it's metaphase. Some people think middle and M for meta as a way to remember it. That's metaphase. B, we've got two nuclei formed in one cell. That must mean we're at the end of mitosis, which means B is at telophase. And then C, we can see those chromatids are forming that very iconic V shape. They're being pulled apart to opposite ends of the cell, which means we're past metaphase, but we're not at telophase yet, which means we're at anaphase. And that will get you 3 marks being able to identify each one. You'll then asked to name structures labeled X, Y, and Z in cell A. So X is pointing at the chromosome. You can call them in this situation actually chromosome or chromatid, it is fine. So that's pointing at the chromosome. Y is pointing at the middle of that chromosome, which remember is a centromere. And then Z is at the fibers, and remember they're spindle fibers. Oops. So that would get you 3 marks as well. So that's a nice 6 marks. That's why it's really important to know all the stages of mitosis really well, really confidently, be able to identify them from images, because you get quite a lot of marks for them. And what we're going to look at now is some practical skills and how we can investigate mitosis. So some of this is sort of a recap of how to use a microscope, but it's really, really important again, and I'll go through that when we get to it, why you need to know how to use a microscope really well when writing about it in the exam. So of the things that you need to know, there's a few techniques that you need to understand when studying mitosis if you were going to study it in a lab. You need to know how to stain root cells on slides, and how to use an optical microscope and a graticule. That's what I'm going to go through now. So the first thing I'm going to go through is how we stain root tips. So root tips can be stained and then squashed to allow us to observe mitosis. So what you need to know from this is how to prepare and stain a root tip in order to observe the stages of mitosis, to basically get images like that. They obviously won't look as nice and neat because they're drawings, but to get images like that from an actual live specimen. Some safety points I'm going to point out now really quick is, if you're ever asked for health and safety for these sorts of questions for this, you always obviously will need to be wearing goggles. We're working with stains and liquids, so goggles. A lab coat also is very sensible, because again, we're working with stains, you can stain your clothes. So goggles, lab coat, and gloves. Otherwise, you can end up being stained a different colour for quite a while. So step-by-step process for investigating mitosis using staining root tips and squishing them. So what you would do is you would cut 1 centimetre from the tip from a growing root. So you might pick an onion, for example. So from the growing root of an onion, you cut 1 centimetre from the tip of this growing root. Really, really important. It needs to be the tip because that is where growth occurs. So that is where my mitosis is going to take place. If you don't pick the tip, you're not going to be able to see anything happening because growth won't be happening. If you're using some stains, you sometimes need to fix them in ethanoic acid as well. So if you were using ethanoauracil stain, you would need to also fix it in ethanoic acid. But generally, this is what you will just do. Cut 1 centimetre from the tip of a growing root. Then you're going to prepare a boiling tube, which is going to contain 1 mole of hydrochloric acid, and you're going to put it in a water bath that's at 60 degrees Celsius. You're then going to transfer your root tip that you've cut into that boiling tube and incubate it for 5 minutes. Then you're going to use a pipette to rinse the root tip really, really well with cold water and then leave the tip to dry on a paper towel. So we're not going to press it, we're not going to squeeze it, we're just going to leave it to dry. So we're removing that hydrochloric acid with a rinse, a cold rinse, and then we're putting it just on a paper towel and leaving it to dry. You're then going to place the root tip on a microscope slide and cut 2 millimeters from the very tip of it. Again, we want to be right at the top of it because that's where that growth is going to be occurring. You're going to get rid of the rest. You can put the rest in the bin. And then using a mounted needle, you're going to break the tip open and spread the cells out really, really thinly. So basically all this process before is basically been allowing us to view these cells and break them up. You're then going to add a few drops of a stain and you're going to leave it for a few minutes. The point of the stain is to make the chromosomes easy to see under a microscope. Otherwise, everything's sort of a clear colour and you can't see anything. There's lots of different stains. I've used the example of toluidine blue O. It's one that you use quite regularly. If you get it on you, you will be stained blue. But then there's also ethanoauracil or Fuelgen stain. If you use Fuelgen stain, you have to also add in an extra rinse. But any named stain is absolutely fine for the exam. You would then place a cover slip over the cells and you push down firmly and squash the tissue. It's one of those few times that we tell you to push them down firmly and squash the tissue. Normally we say don't squash something, you won't be able to see anything, but for these we want to squash the tissue. The reason behind this is it makes the tissue thinner and it will allow more light to pass through it. We don't want to smear the cover slip sideways because you'll damage the chromosomes, or your chromosomes will get moved and you won't be able to see what stages of mitosis is going through. Once you have done that, you can now look at all the stages of mitosis under an optical microscope. And you should see something like the picture I've got on this slide. It regularly doesn't happen and doesn't often look like that, but you will often get pictures like this in the exam and you're asked to point out the different stages. So if we look at this, we can tell this is interphase for this one because the cell's not really very condensed. It's not a dark color, it would be more condensed if we're going through mitosis. Just one whole cell, one nucleus, that's it. So interphase, prophase. So we've got those chromosomes condensing in this situation and the centrioles forming. Prophase here, we can see that that's prophase because we've just got this big mishmash. We've got no nuclear envelope and this big mishmash of all our chromosomes that are really, really fat in the middle. Metaphase, we can see actually them nicely lining up along the equator of the cell. Anaphase, we can see that V shape, they're getting pulled to either pole of the cell, and then telophase, you can see that they're starting to split here and start to form 2 separate cells. What we're going to do now is we're going to just have a quick overview of the stages of using a microscope. Now, this seems really, really basic and really, really silly to go through at A level. However, you regularly get asked questions on this. And if you don't do it in the right process and say how you would use a microscope, you lose lots and lots of marks on these questions, especially if there's one that asks you to explain this whole process. And explaining this process of how you might observe mitosis and root tips in a lab would want you to explain those steps we've just gone through on the previous slide, and then how to look at it through a microscope. Really, really, really important. Clip your slide you've prepared onto the stage, but your important part is you always start on the lowest powered objective lens. And that is the one that produces the lowest magnification. So you want to start at it from a lower magnification so you get an overall view, the overall image of what you're looking at. So then you can pick areas you maybe want to focus in on and magnify more. So then you can look in closer at different sections, but you want to have a big overall view of what you're viewing to start off with. Sometimes you don't actually need to go to a higher magnification. So that is why we say you start at a low one and then increase your magnification if needed. So you start at your lowest powered objective lens. You then use your coarse adjustment knob to bring, I wrote objective knob, I mean coarse adjustment knob, to bring the stage up just below the objective lens. Again, keep your eye on that because I've seen people before, you bring it up too high and then you crush the glass and everything gets ruined. So just below the objective lens, not onto it, not touching, just below. So if you need to, don't be looking down the microscope at the time, look at yourself doing that. Then look down the eyepiece, because you don't need to look down the eyepiece until this point. And your eyepiece contains the ocular lens. Once you're looking down the eyepiece, use your coarse adjustment knob to move the stage downwards away from the objective lens until the image is roughly in focus. And again, I emphasize downwards. You do not need to bring it any higher. You've got it as high as you need, and then you're going to bring it down to try and get the image roughly in focus. Then, to make it more in focus, you're going to adjust your focus with the fine adjustment knob. And that is until you get a clear image of what's on the slide. If you need to see the slide with greater magnification, you are then going to swap to a high-powered objective lens and refocus using those same processes. If at any point in an exam you're asked to draw cells undergoing mitosis or to talk about this, you make sure you write down the magnification the specimen was viewed under. So that's really, really important. Mitotic index. So mitotic index is the proportion of cells that are undergoing mitosis. So you've got your root tip, you've squished it. You've viewed the cells under a microscope and you've worked out how many are in metaphase, how many are in anaphase, how many are in telophase, and how many are in interphase and not go through mitosis. So you can calculate the mitotic index of your cells using this formula. So number of cells with visible chromosomes divided by total number of cells observed. Ones without visible chromosomes are ones that will not be going through mitosis. This lets you work out how quickly the tissue is growing. Basically the idea is the more number of cells with visible chromosomes, the faster mitosis is happening compared to the number of cells observed in total. And it lets you know if anything's going on that shouldn't be. So a plant root tip in this example is constantly growing. So you would expect to have a high mitotic index, which means lots of cells going through mitosis because it's constantly growing. So it should be going through mitosis highly. If you were looking at a different tissue sample, a high mitotic index could suggest that the tissue is going through repair in that location, or it could even indicate that there's cancerous growth in that tissue. So that is why mitotic index is useful. You're going to expect it to be high with root tips because that's an area that's going through growth. Another reason it could be high is due to repair or cancerous growth. Finally, you need to understand what a graticule and a micrometer are. These help us get the size of cells. So if we're looking down a microscope, we need to know the size of the cells that we're actually looking at rather than just going, oh, they're this big. So you need to be able to calculate the size of the cells you're looking at. And this is the eyepiece graticule and stage micrometer come in. And they're basically like rulers. So the eyepiece graticule is fitted onto the eyepiece. So here on your eyepiece, you'll have an eyepiece graticule, which is fitted onto there. It's like it looks like a transparent ruler with numbers, but it doesn't have any units on. So you'll be able to see that. And this is your eyepiece graticule. It will look like that when you're looking down the microscope. And you can also have a stage micrometer. That is placed on the stage, and it's a microscope slide with an accurate scale. So we can pop this on top of the slide. So this one has units on it. And what we do with this, it's used to work out the value of the divisions on the eyepiece graticule at a particular magnification. So what this means is when you take the stage micrometer away and replace it with a slide containing your tissue sample, you're able to measure the size of the cells. So what you do is you line up the eyepiece graticule and your stage micrometer. So you line them both up at 0. Each division on your stage micrometer is 0.1 millimeters long. At this magnification, one division on the stage micrometer is the same as 4.5 divisions on the eyepiece graticule. So to work out the size of one division on the eyepiece graticule, you just need to divide 0.1 by 4.5. And that would give us 0.022 millimeters. So if you look at a cell under the microscope at this magnification, and it's 4 eyepiece divisions long, you know it measures 4 times 0.022, which equals 0.088 millimeters. You need to, this is really, really important, recalibrate your eyepiece graticule at every different magnification. So if you're changing magnifications, you need to recalibrate it every time because it will change. Or if you are given an image of cells in the exam, so if you're given an image of cells under a microscope in the exam, you can calculate their actual size using this formula. You'll know this formula well from GCSE. Actual size equals your size of the image divided by magnification. So for example, if you were given an image of a cell that measures 5 millimeters and the magnification was times 100, then the actual size of the cell would be 5 divided by 100, so our actual size would be 0.05 millimeters. Couple of other things to go through when looking down a microscope. Artifacts. Artifacts are anything that you can see down a microscope that aren't part of the cell or specimen that you're looking at. So that could be dust, air bubbles, fingerprints, anything that you've caused by squashing or straining your sample. They usually occur during preparation of your slides and shouldn't really be there at all. So you have to prepare, for example, in this situation, your root tip carefully to avoid creating artifacts. Electron micrographs, actually, you get lots of artifacts in them because the specimens have to go through a lot of preparation before you can view them under an electron microscope. And the first scientists to use these microscopes could only distinguish between artifacts and organelles by repeatedly preparing specimens in different ways. And if an object could be seen with one preparation technique but not another, it was most likely to be an artifact than an organelle. So a question. A sample of cells was prepared to observe mitosis. In total, 42 cells were observed. 32 of those had visible chromosomes. And you're asked to calculate the mitotic index for this sample and give your answer to 2 decimal places. So all you need to do is you just need to do 32 divided by 42. When you do 32 divided by 42, that would give you 0.761904761. That'll get you 1 mark. Second part of the question asks you to give your answer to 2 decimal places. So then your answer would be 0.76. What we're going to look at now is just a few more exam questions on mitosis. I just want to go through a couple of exam questions now. So question 1. In this one, a student investigated mitosis in the tissue from an onion root tip. I'd highlight the word mitosis and onion root tip, just to let me know, but we're probably talking about the root tip squish experiment and we're looking at mitosis. So the student prepared a temporary mount of the onion tissue on a glass slide. She covered the tissue with a cover slip, and she was then given the following instruction. Push down hard on the cover slip, but do not push the cover slip sideways. Explain why she was given this instruction. So it's 2 marks. So we'll look at the instruction as 2 parts. So push down hard, we're going to look at first. So why was she told to push down hard? Well, we need to push down hard to squash the tissue. We need to actually squash the tissue. So she was told to push down hard to spread and squash the tissue. That get you the first part of the mark. The second part of the mark is going to come from, but do not push the cover slip sideways. So why don't we push the cover slip sideways? Let's have a look at that. Well, we don't push the cover slip sideways. So not to push sideways, not push sideways. And we don't push sideways. And that is to avoid rolling the cells together, breaking the chromosomes. So that question would get you 2 marks. First mark for explaining why we don't push down hard. Second for saying we don't push sideways. And we don't push sideways to avoid rolling cells together and breaking the chromosomes. So the image below, now, question continues, shows one cell that the student saw in the onion tissue. From this image, the student concluded that the cell in the image above was in the anaphase stage of mitosis. Was she correct? Give 2 reasons for your answer. So saying yes or no does not get you any marks. What you need to do is actually justify what you are doing, what you are saying, and what they are saying in this process. So let's have a look at this. What is happening? So the chromosomes are in 2 groups and they've got the V shape. So let's have a look. So we know anaphase, the centromeres are dividing and this sister chromatids are being pulled to opposite ends of the pole. And we have that V shape, and we can see that V shape really, really nicely there. So what we're going to write here is we're going to put, was she correct? Give 2 reasons for your answer. I'm going to pop yes here and then say why. Our chromosomes, our chromatids, in this situation, I'm going to talk about them as chromatids. So our chromatids are in 2 groups at opposite poles. At opposite poles of the spindle. So that at opposite poles of the spindle. So that will get you 1 mark. Another reason we've got that nice characteristic V shape. So we've got the V shape. We need to say why the V shape is important. So the V shape shows that the sister chromatids have been pulled apart at their centromeres. That'll get you a 2nd mark. So the student counted the number of cells she observed in each stage of mitosis. Of the 200 cells she counted, only 6 were in anaphase. 1 cell cycle of an onion root tissue takes 16 hours, and it asks you to calculate how many minutes these cells spend in anaphase and asks to show your working. So let's have another quick look. So she's got 200 cells, only 6 were in anaphase. So that's 6/200. Of the onion root tissue, one cell cycle takes 16 hours. Calculate it in minutes. So let's get 16 hours in minutes. So 16 times 60. Would be 960. And so then we've got the 16 hours. We just need now 6/200 of 960. So that would be 6/200 times by 960. So that would be 0.03 times that by 960. And that's going to give us 28.8 minutes. And you can round it up to 29 if you want. Either is correct for this answer, and that would give you 2 marks, a total of 6 marks for that question. So the figure below shows some cells from an onion root tip at different stages of the cell cycle, and it asks you here to place stages A to E in the correct order and to start with D. So we're told D is the beginning of the cell cycle. So then it wants us to put the rest in order. So we've got D. So the next phase, we're going to have that condensing of chromosomes. So we can clearly see the chromosomes are condensing here at C. So it's going to go D, C. Once the chromosomes are condensed, they're going to line up along the middle of the cell for anaphase. So we've got prophase, metaphase, anaphase. So we've got metaphase next, lining up along the middle. M for middle, metaphase. So B. Anaphase, they're going to be pulling apart to opposite ends. So B. And then finally, you can see we've got 2 nuclei forming in 2 cells. A. That will get you 1 mark. So to obtain these images, the onion root tip was cut off, stained, and put on a microscope slide. A cover slip was placed on top. The root tip was then firmly squashed and viewed under an optical microscope. Complete the table below to give one reason why each of these steps was necessary. Taking cells from the root tip, remember, because that is the region where mitosis occurs. Region, mitosis occurs because that's where that growth is happening. Firmly squashing the root tip, that is to allow light through. Or to make the tissue layer thin. So figure below shows how the amount of DNA per cell changed during interphase and meiosis in an animal. Explain how the behavior of chromosomes causes these changes in the amount of DNA per cell between F and G. I'm going to go through this, but obviously we've been looking at mitosis, but it does sort of cover it. So at F, clearly what's happening between F and at 4, our DNA is going to be replicating. So at F, our DNA replicates. So it gets up to here and it's replicated. Then our homologous chromosomes are going to separate. So that's going to be our second decrease of our first decrease, sorry. So our homologous chromosomes separate. And then our sister chromatids separate. So what would happen to the amount of DNA per cell at fertilization of cell G? DNA would double. Just going to skip past this one. And I'm going to leave the exam questions there. So this was the topic on mitosis. Thank you for watching.